Okay.
All right. So we're okay. So, but I really wanted to have you describe for people who are going to be watching this, like how you actually got into circadian biology, right? It was like you came from a neuroscientist perspective, right? Um,
I
is that
actually,
correct? And can you kind of just trace what your evolution was to become a circadian biologist?
sure. It's going to be a little bit of a long answer, but I'll do it fast.
No, no, it's fine.
I come originally from Jordan in the Middle East, and
Mhm.
I was born Catholic there, 2% of the population.
Amazing.
And, I just never thought of science, which is humbling when I see people who say, I knew I'm a scientist. I was, dissecting animals when I was 5 or 6. And I'm like, was playing soccer and never thought
Uh-huh.
of being a scientist. And then I saw the genetics class. I was devout Christian Catholic, and then I was like, oh wow, look at this guy, this priest Mendel, this is great.
Ah,
So I could be, and I was thinking of going to priesthood. So I was like, oh, I could be priest and a scientist. So I really went into science by way of religious upbringing.
wow, that's amazing. Okay.
And
Uh-huh.
so then, I did very well in my last high school grades, and I could have gone into what Jordanians like, medicine, lawyer, or engineering. But I decided to go to biology to a small university at north of Jordan, and it took me a long time to make it to the US to do my studies. First, I started working on learning and memory, but once I got heard about circadian rhythms, this is when I went into circadian rhythms. So it's a long story, but I wasn't literally determined to go into circadian rhythms at the beginning.
No, so but at the same time, so what actually like drew you into this particular question? Because in fact, you've actually found a new photoreceptor, right? There's not rods or cones that the rest of us plain biologists are aware of, right?
Yeah.
But you've actually, discovered a new photoreceptor that
Right.
actually contributes to circadian biology. So,
Right. So I actually worked 7 years and a half for my PhD for circadian biology in aplesia first, and
mhm.
I didn't do any groundbreaking research in circadian rhythm until I moved to the mouse, as you said, and
Uh-huh.
I was working on the eye of aplesia, and this is where it connected me to the mammalian eye. And then when we discovered the IPRGCs, the new photoreceptors that are non-rod, non-cone, this
Right.
really puts me
With
on
the
the,
multiple isoforms, right?
yeah, with multiple
So, right,
subtypes.
right.
Yeah, 6 different
Yeah.
subtypes now.
Right.
It's crazy.
Oh, it's six now. I thought four, five. Right, but
Yeah.
okay.
Five was just a few years
Okay.
ago. We find a new
Yeah.
one every other year.
Okay.
Yeah.
Wow, that's amazing though.
Yeah, it is pretty remarkable that not only we miss them, but the fact that they are so diverse and contribute to so many behaviors that I would love to discuss if we have time.
No, we do have time. Please, can you tell us about this? In fact, in terms of the different isoforms, I'm particularly curious about like how, in fact, each one of them is contributing to like perhaps how they're working on the optic nerve, or like how they're impinging, I guess, on the optic nerve.
I have to say, this is something, again, I would love when I was looking for a job, and even in some of my Hughes interview, when I was invited to apply for the Howard Hughes, a lot of people would say, so you're only going to work on IPRGCs? Is there anything else to discover? And honestly, I don't take this as a negative question, because at the time we thought they were circadian photoreception. And circadian photoreceptors, when we published this work in 2002, we actually
Mhm.
called them circadian photoreceptors. But
Right.
we quickly found that they influence many other functions beyond the circadian clock. And as you said, what's the function of the different subtypes? I think the best function of a subtype that is really cool right now is the M4 population, done by one of my postdoc who has her own lab right now at Northwestern University,
Oh,
Tiffany
okay.
Bennett.
Mhm.
She found that they are actually your classical contrast detecting ganglion cells, and that melanopsin signaling in these cells allow them to signal contrast much better if melanopsin is there versus not there. So it's really cool how it evolved from circadian photoreception all the way to contrast detection.
Oh, that's really cool. But is M4 then the only receptor that is actually susceptible? Well, sorry, actually requires melanopsin, or
No, it's
when you've done
actually,
the knockout before, have you seen any of the other subtypes also
So this is really a great question. And so the M4 cells, they were completely studied very much in the retinal field. So they were these on-alpha ganglion cells. On-alpha ganglion cells are known to be important for contrast detection. But we discovered that they have melanopsin. And people said, oh, maybe this is an evolutionary artifact, that why would you need melanopsin in a contrast detection ganglion cells? And then Schmidt showed beautifully that you actually need melanopsin in these cells that usually function to measure contrast from rods and cones and outer retina. They still need melanopsin. So that's really cool. So for us, one of the biggest problem we had is that people always ask, what's the function of melanopsin if these are ganglion cells and they could receive rod and cone input? And we have still unpublished work, but we have some indications that these are very important for detecting light that is available for a prolonged period of time. Rods and cones are good at measuring light for shorter period of time, while the melanopsin system measure lights for very prolonged periods of time. So we think partly is that melanopsin is there to measure seasons, and circadian clock are important for annual clock. So we think that animals without calendars, they are not like the plants, just can measure, photoperiod very easily because they are always exposed to light. They have to develop method to know when the seasons are going to happen, so they could be ready for the harsher winter months, for example.
But again, these are mammalian specific, right? So it's like, and do you find anything similar in other organisms?
They are not mammalian specific. That is the.
Oh, I thought they were. Okay, sorry. That's my mistake.
So the difference between, in mammals, that these photoreceptors are all centralized to the retina. In non-mammals, you still have the IPRGCs. In fact, I published work with Marnie Halpern on zebrafish in the eye, but they have extraocular photoreceptors as well.
Okay, and so they're just located in a different part of the brain.
Yes, and the extraocular, different part of the brain or even different organ. Like, so for example, zebrafish, the kidneys are light sensitive. So and they may use different opsins. So why was melanopsin chosen specifically for these human IPRGCs, for the mammalian IPRGCs? That I don't have a clue.
And the melanopsin, exactly what wavelength does it actually receive?
So melanopsin is best at 480, but opsins are known to have a wide range of responses. So all our opsins respond to a range of colors. They don't just respond to a specific color, which is good. Otherwise, we would be in problem if they responded very specifically to a single color. We would have missed a lot in the environment. And melanopsin was first identified in the frog melanophores, in the skin of the frog. Hence the name melanopsin, melanocyte opsin.
Okay, yeah, that makes sense. So, but in fact, can it respond to light that actually transperce the skull, or no, or does it require any kind of like optic reception?
Yeah, so for us, for humans, melanopsin is all in the eye. There is evidence it may be in the pupil muscle, but I'm not, I'm not completely convinced. So I think for us, mostly it's through our eyes. So that's why if we don't have our eyes, we suffer tremendously as, all our entrainment, all our light reception through the melanopsin system happens through the eyes. So everything is centralized in mammals. We're not like the non-mammalian.
So it's not like the kind of like blue light that we get through the skull or whatever. It's like that.
Honestly, blue light is hard to transverse so much into the skull. Usually longer wavelength light can cross more. But even if you think of the beautiful bird experiment by the great Mike Menaker, which we lost recently, unfortunately, they have to shave the hair of the birds on the top to allow the pineal gland to respond to lower light intensities. So even with birds, if you want at lower light intensities, that not very high light intensities, you have to shave the hair. And only if they put a little bit of a black paint, they blocked all the responses from the skull. So the eyes are different. They are much easier to get light through the eyes than.
So can I ask also, I'm just curious, do you see any like sex-specific effects of melanopsin?
That's a great question. Honestly, we don't have anything published about this area, but I always wonder. I mean, there are really major differences in sleep between men and women, and I wonder if there's some contribution to the way we sense light. So that's something that would be interesting to look in the future. We don't have any specific answers for this right now.
Okay, but just nothing yet. Yeah.
Nothing yet.
This sounds really
cool. Yeah. Nothing
Sorry.
that we know of. Yeah.
Okay. No, it's a fascinating subject, and I really, really appreciated your talk. And yeah, thank you so much. We've had so much COVID-19. I was so happy to actually see some talks, and I really am very, very happy to Cold Spring Harbor for bringing this together. And thank you so much for taking time to speak with me now.
It's my pleasure. Absolutely. My pleasure. Thank you so much.
Well, okay. I hope to see you again in future.
Great.
For real, face to face. Okay, done.
Thanks a lot. Thanks for the invitation.